Hello students, today we are going to discuss about Operon concept. This is one of the important topic in 12th biology. So what is Operon? If you are able to recall in your theory classes, Operon is a concept which explains about the lactose metabolism in bacteria. Operon was first time explained by the scientist called Jacob and Monod, and they shared Nobel Prize in a medicinal field. So before we go to the Operon, let us understand how many terms in the Operon. Look at there are two terms are there. The first term called lac, which represent lactose disaccharide sugar, and this lactose disaccharide sugar is made up of two monosaccharides. One is glucose and other one is galactose, and this glucose and galactose are bound by the glycosidic bond, right? And the other term called operon. So what is the meaning of operon? Actually, in a simple term we can say operon is a small segment of DNA. A operon is a small segment of bacterial DNA, right? So what is the exact definition of operon? Functional unit of DNA contains cluster of genes which are regulated by what? One single promoter, right? If you look at that operon, a small segment of DNA in bacteria, prokaryotes, this entire operon in bacteria has been divided into two regions. The first region called structural gene region, the second region called regulatory gene region. First region is a structural gene. Structural genes are three different genes are present in the structural portions. The first look at the structural gene region. Structural gene region has three genes, structural genes. It has three structural genes. The first structural gene is called LAC-Z, and this LAC-Z is code for the beta-galactosidase enzyme. This LAC-Z gene undergo transcription and translation to produce beta-galactosidase enzyme, and this enzyme is essential to break the bond between glycosidic bond which present between glucose and galactose. And the other structural gene, the second structural gene called LAC-Y. LAC-Y gene is code for the permease enzyme. So undergo transcription and translation to form an enzyme called permease. This permease enzyme which allows lactose from external medium to inside the bacteria. When permease enzyme is present in a bacteria, it increases the permeability of plasma membrane to lactose. So when permease enzyme is present in bacteria, it allows more and more lactose from external medium to inside. And the last gene in the structural portion we call LAC-A gene. LAC-A gene is code for the enzyme called transacetylase enzyme. And this LAC-A gene undergo transcription and translation to produce the enzyme called transacetylase. This transacetylase enzyme which can transfer the acetyl group from acetyl-CoA to beta-galactosidase. So these three genes are known as structural genes in the operon. Look at the other portion of operon we call regulatory genes. Regulatory genes are again classified into three groups. Look at the first one. At the end of the operon, the first gene called promoter gene, represented by capital P, promoter gene. And the other gene we call I gene. I gene is code for the, and the I gene is responsible for the synthesis of protein called repressor protein. So this I gene undergo transcription and translation to produce repressor protein. And this repressor protein is high efficient towards the operator region that we will discuss a minute later. The next gene we call operator gene. Students, keep in your mind, in the regulatory gene region in an operon, the operator gene work like a switch. So everyone keep in your mind, the operator gene can turn off and on the structural genes. That is the reason why whenever you're reading, whenever you're trying to understand the lac operon, everyone need to understand one more important concept, that is called regulatory genes are control the structural genes. And which part of the regulatory gene in an operon controlling the structural genes? Operator gene. Operator gene work like a switch. It can turn on the operon and it can turn off the operon. Am I clear? And moreover, keep in your mind, today we are going to understand two scenarios. Number one, in presence of lactose and absence of lactose. Right? So let me explain what would happen when the lactose is absent in the medium. How this operon is going to be turned on and off. And everyone keep in your mind, students, lac operon in Escherichia coli bacteria, 90% of time it will be in the form of a off mode. Because Escherichia coli bacteria always try to absorb the glucose. If glucose is not available in a medium, when large amount of lactose is available in a medium, that time structural genes will on. When abundant glucose is present in a medium, when there is no lactose available in a medium, in that time all the structural genes will become off mode. Structural genes in the form of off mode. So this is one of the inducible operon. What is meant by inducible operon? Most of the time the operon is in the form of off mode. When requirement comes, the structural genes become a on mode. Such a type of operon we call inducible operon. From off, it turned to on. Understand the first scenario. When there is no lactose in a medium. Look at here. What will happen when there is no lactose in a medium? I hope you all are following me because this is an important concept. When there is no lactose and abundant glucose is available in a medium, bacteria can turn off the structural portion. How they can turn off the structural genes? Look at here. Whenever lactose is absent in a medium, regulatory I gene, LAC-I gene, which is code for the protein called repressor protein. Do you remember I told you? The I gene is responsible for what? Repressor protein. So this I gene continuously undergo transcription and translation to produce repressor protein. This repressor protein is high efficient towards the operator gene. So this repressor protein go and binds to the operator region of regulatory genes. So when the repressor protein go and bind to the operator gene, there is an enzyme called RNA polymerase enzyme which attaches to the promoter region. Look at the promoter. This is the promoter region. So to the promoter region, which enzyme has been come and attached? RNA polymerase. If you able to recall transcription process in the theory classes, RNA polymerase enzyme is essential for the initiation of transcription. Yes or no, student? So where is our RNA polymerase enzyme? RNA polymerase enzyme on the promoter region. Most of the students feel too difficult to remember this. Whether the RNA polymerase enzyme is attached to the operator or promoter. Most of the students get confused whether the RNA polymerase enzyme is attached to promoter or operator. It could be easy to remember while you're reading. Just remember the two P's. First P in the polymerase enzyme and the other P a promoter. So if you remember the two P's, it could be easy to remember, it could be easy to understand. So where is our RNA polymerase enzyme? RNA polymerase enzyme on the promoter region. So whenever repressor protein go and bind to the operator region, in that time operon will switch it off. Why it's going to be switched off? Because this RNA polymerase enzyme cannot slide over the structural genes. This RNA polymerase enzyme cannot read what's written on the structural gene, because promoter region, on the promoter region, RNA polymerase enzyme is there. I told you that RNA polymerase enzyme cannot slide over the structural genes, because look at the operator gene. Operator on the operator gene, repressor protein, it has blocked that pathway, right? Because of the repressor protein blocked the operator, our RNA polymerase enzyme cannot slide over the structural genes and it cannot transcribe the structural genes. So when the RNA polymerase enzyme cannot go forward, LAC-Z, LAC-Y, LAC-A enzymes, LAC-Z, LAC-Y, LAC-A genes cannot transcribe and translate to produce three different type of enzymes. Now, what makes the LAC operon turns on? So, if you take another scenario where the lactose is present in a medium, glucose is absent, that time lactose, small amount of lactose initially entered into the bacterial cell, and that lactose convert into another form called allolactose. So allolactose work like a inducer. So this allolactose is high affinity towards the repressor protein. So where is our repressor protein? If you are able to recall, repressor protein is bind to operator region, right? When the repressor protein is bind to the operator region, structural gene has been turned off, right? So but when the lactose is entered into the bacterial cell, initially very little amount of lactose is entered into the bacterial cell, it convert into the other form called allolactose. Allolactose is high affinity towards what? Repressor protein. So allolactose go and bind to the repressor protein. Now repressor protein and allolactose are free, which means our operator gene is free. In this condition, RNA polymerase enzyme, where is the RNA polymerase enzyme? On the promoter region. Right now, the RNA polymerase enzyme slide over the structural gene and they transcribe to form a various enzymes. Now, RNA polymerase enzyme slide over the structural genes. Now, all the structural genes called LAC-Z, LAC-Y and LAC-A undergo transcribe and translate to form a various protein that are participate in the metabolism of what? Lactose. So when you take, students, keep in your mind, when the lactose is available in the medium, structural genes are going to on. So how they're going to on? One more time, I'll explain you. Whenever initially a little amount of lactose is entered into the bacterial cell, it turns into other form called what? Allolactose. So that allolactose is high affinity towards what? Repressor protein. So where is the repressor protein? On the operator gene. So now when the inducer or what? Allolactoses go and bind to the repressor protein, so the repressor protein release or remove from the operator gene. Now the operator gene is free. Now in this condition, RNA polymerase, which was attached to the promoter region, slide over the structural gene and they transcribe all the structural genes to form what? Different mRNAs and different enzymes. So this all the enzyme I told you earlier, beta galactosidase enzyme can break down the bond, glycosidic bond between glucose to galactose. And permease enzyme, remember here, look at here, the permease enzyme. Permease enzyme, when it increase in a bacterial cell, it allows more amount of lactose from outside to inside. I told you initially less amount of lactose is entered into the bacterial cell, but once the permease enzyme concentration is increasing in the cell, it allows the more amount of lactose from outside to inside. I told you it is a tube-like protein which is bind to where? Plasma membrane. And I told you the very important point here, what? Permease enzyme can increase the permeability of bacterial plasma membrane to lactose. The last enzyme is called what? Transacetylase enzyme. And what is the function of transacetylase enzyme? It transfers the acetyl group from acetyl-CoA to beta-galactosidase. So when the operon is turned on, all the structural genes are going to transcribe and translate to produce a different enzymes, and that enzymes are participating in the digestion of what? Lactose into glucose and galactose. Am I clear, student? And moreover, everyone keep in your mind, at the end of this video I wanted to tell you something. Lac operon is a one type of polycistronic operon. So what is the meaning of polycistronic? If you take cistron word, it is a small segment of DNA. Do you remember at the beginning of the class I told you operon is a small segment of DNA which consists one promoter and operator. If more than one polypeptide chain or more than one protein synthesis genes are present, we should call polycistronic. If look at the lac operon, at the first end what? Promoter region. At the last end what? Terminator region. So how many protein synthesis genes are present here? 3 protein synthesis genes are present, right? Lac Z, Lac Y, Lac A. So shall we call this a polycistronic? More than one polypeptide chain or more than one protein synthesis genes are present. So it is called polycistronic. In bacteria, operon is polycistronic operon. But if you look at the eukaryotes, in a eukaryotic DNA segment in a cistron, small segment of a DNA, between promoter to terminator, one protein synthesis gene is present. So eukaryotic DNA is called what? Monocistronic. But prokaryotic or bacterial operon is called what? Polycistronic. So I hope you understand when lac operon is going to off and when lac operon is going to on. I hope you understand what is lac operon and when it is going to turn off and when it is going to turn on. Thank you very much.